Okay, welcome back. So in the first part of my talk, we have discussed some very basics of what DNA is and then how to do DNA analysis and how DNA analysis is and what technologies are available for different kinds of sequencing and other DNA analysis. So in my second part of the talk, I'm going to talk about the genetics of cancer. Because I'm a cancer geneticist and we work a lot on genomics of cancer diagnosis and early detection. So cancer is basically a genetic disease. On all cancers, they occur because of mutations in some genes, and based on the kind of the cancer, it can be divided into sporadic, familial, or hereditary cancers. So, as I said, cancer is a genetic disease, so all cancers come under a genetic disorder. And then sporadic cancers include for more than 70% of all cancers. So what I mean by sporadic is that mutations in certain cancer predisposition genes, they accumulate over the course of our development. So these can be because of external sources or like chemicals or radiation or other factors. Or it could also be internal, where we have mutations in, we inherit some mutations in some genes, and then they additionally cause other mutations in cancer predisposition genes. So most of the cancers are sporadic, and there are 15 to 30% of cancers that are familial. So these are cancers that run in families, but there's no pinpoint accurate estimation of what the mutation or what the factor that causing cancer is. And then there are hereditary cancers that account for 5% of all cancers. So these are so these tend to be much earlier on in onset. So this could be childhood cancers or early development of breast or colorectal cancers. So what is cancer? So cancer is a more virulent form of neoplasia. So in all types of cancer, so in all types of cancer, there are some of the body cells begin to divide uncontrollably and then spread to surrounding tissues. And then cancer is caused by changes or mutations in genes that control the way our cells function, especially how they grow and divide. And as you all might have known somebody in your families or friends who have cancer, cancer can start almost anywhere. So there are more than 100 types of cancer in different organ systems and organs or tissues. And for a new growth to become a cancer or neoplasia to become a cancer, it must be malignant at the primary site and metastasize so that is spread to more distant sites. So here you can see there's a normal epithelium. There is uncontrollable cell division or proliferation. Then there is an invasion in the local tissue. And most cancers, they also invade the local lymph nodes. And sometimes they also go to distant metastasis. So they go on to affect other organ systems. So there are three main classes of cancer. One is carcinoma, sarcoma, and lymphoid neoplasms. So this is hematopoietic and lymphoid, where the cancer spreads through bone marrow, lymph, or the peripheral blood. And within each of these groups, tumors are classified by the site, the tissue type, the histological appearance, and the degree of malignancy. And then most of these cancers, they exhibit chromosomal aneuploidy. So they don't usually have 46 chromosomes like the normal cells do. And then they're also characterized by numerous mutations within the tumor. So according to the National Cancer Institute of the National Institute of Health, United States, the cancer is classified into carcinomas, sarcomas, myeloma, leukemia, lymphomas, and the mixed type. So carcinomas, they basically refer to malignant neoplasms of epithelial origin. So these are cancers of the internal or external lining of the body, and these account to 80 to 90% of all cancer types. So even carcinomas can be divided into two major types. One is adenocarcinoma, which develops in an organ or a gland. And then there is squamous cell carcinoma, which originates in the squamous epithelium. And then sarcoma basically refers to cancer that originates in the supportive and connective tissues, such as bones, tendons, cartilage, muscle, and fat. And these generally occur in young adults, and most commonly, sarcoma often develops as a painful mass on the bone. Sarcoma tumors usually resemble the tissue in which they also grow. So myeloma is cancer that originates in the plasma cells of the bone marrow. And leukemia are also cancers of the bone marrow. And then lymphoma develop in the glands or nodes of the lymphatic system. And then there are some rare cancers that are also mixed type. Some of them are adenosquamous carcinomas. So these are mixed mesodermal tumors, and then there are carcinosarcomas. So there's not, sometimes it's very hard to delineate at what site these cancers occur, or what kind of tissue these cancers have occurred from. So that's when these are the mixed types. So most cancers are divided into, classified into by TNM staging. So T is called the tissue or the organ where the tumor goes. N is to show whether it has invaded a local lymph node. And M is to show whether there was metastasis or not. So based on the stage, so you can actually, the TNM actually gives you more information about the clinical characteristics of the cancer. So for example, if I say there is, the cancer is T4, N1, and M1, so it means that T4 is, it has invaded adjacent structures. And then there is N1 is, there are few nodes involved. And then M1 as in there is distant metastasis, so there is cancer in other parts of the body as well. So let's see what causes cancer. So is it a genetic disease? Yes, it is a genetic disease, and based, as I just discussed, there could be germline mutations or somatic mutations. So germline mutations have occurred in the germline. So these mutations would have occurred in the parental germ cells, and then the patient would have inherited them. So the patients are actually born with these mutations. But somatic mutations are mutations that occur over the life cycle of the individual. So most cancers are they have somatic mutations. So cancer can be caused also by environmental agents, and there are numerous studies to show that alcohol, tobacco smoke, and exposure to chemicals and radiations like carcinogenic chemicals and radiations cause cancer. Can infections cause cancer? Yes. So HIV, which is human immunodeficiency virus, that was actually one of the first few patients that where HIV was discovered was patients who are these homosexuals in the United States that came up with these Kaposi sarcomas, and that actually led. That was where a lot of research on HIV was occurring. And then HPV, which is human papillomavirus, the infections are associated with cervical cancer, head and neck cancer, and a number of anogenital cancers. So EBV, which is Epstein-Barr virus, has been associated with nasopharyngeal carcinomas. And there are other viruses like HBV, HCV that also are associated with other cancers. And then can obesity cause cancer? Yes. So excess visceral fat leads to low oxygen, and then increased inflammation, and then that leads to increased insulin production. And then increased insulin production leads to high levels of estrogen, and then that can cause proliferative cell growth, and that might lead to tumors also. So there are different kinds of mutations that occur in cancers. One is the driver mutations. So these are mutations that occur in genes that are involved in development and progression of cancers. So these genes are called driver genes, and these mutations are called as driver mutations. So one of the most commonly mutated gene is the P53 gene, or the TP53, which is mutated in a vast majority of cancers of different types. And then most mutations are found either through sequencing, appear to be random, or as the cancer develops. So these are called as passenger mutations. So based on the kind of mutations occur in different genes, the cancer predisposition genes are classified into proto-oncogenes and tumor suppressor genes. So these proto-oncogenes have to be activated by mutations to give rise to cancer. So most of these mutations are gain of function mutations, amplifications or point mutations, and promoter mutations that turn the switch on. So these proto-oncogenes become activated oncogenes, and then these oncogenes can give rise to tumors. There are also tumor suppressor genes. So these tumor suppressor genes, they encode proteins that are involved in various aspects of cellular functions and DNA repair. They activate apoptosis upon stress, and then loss of these mutations in these genes are always characterized by loss of functions of both the alleles, or if there's a dominant negative mutation of one allele. So I would all advise anybody who's interested in reading more about the genetics of cancer to go to this paper that was published in Cell 2011 by two scientists, Hanahan and Weinberg. So they've actually determined the hallmarks of cancer, like what is it required, what kind of mutations are required, and what are the different pathways. Excuse me. What are the different pathways that are affected in cancers? So there are 6 major hallmarks. So one is sustaining proliferative signaling. So for a cancer to divide, it should always maintain a turnover of cells by cell division. The other one is evading growth suppressors. So whenever there is unscheduled cell division, there are these growth suppressors that try to stop it, and the tumor should try to evade it. And then activating invasion metastasis, that is one of the hallmarks of cancer. So it should go and able to divide in other tissues and organs. They should also enable replicative immortality. So the cells should be able to divide unconditionally and without any without the replication being stopped. It should be able to induce angiogenesis. So there should be new blood vessels, because these tumors also need nutrition and oxygenated blood. So that is what angiogenesis factors do. So they create new blood vessels in the tumors. And they should also be able to resist cell death. Like there are numerous programs in the cell that allow the cell to either go into necrosis, apoptosis, or necrosis. So these cells, the tumor cells, should be able to evade that. In addition to that, there are emerging hallmarks. So nowadays, everybody's talking about immunotherapies for cancer. So these tumors should also avoid immune destruction. Also inflammatory markers. And then one of the hallmarks is genomic instability. So as I told you before, these cells do not have 46 chromosomes. So there will be a lot of genomic instability, a lot of mutations, rearrangements, and translocations. And then they should be able to deregulate cellular energetics. So based on the different hallmarks, their researchers have also designed different drugs based on which hallmark they want to target. So we all know there's immunotherapy. So these are immune activating antibodies. There are telomerase inhibitors, so that inhibit cell division, so that the cells don't replicate indefinitely. There are anti-inflammatory drugs that that stop tumor that promote inflammation. That discontinue inflammation. They are inhibitors of metastasis and angiogenesis. They are PARP inhibitors that actually increase genomic instability by a lot so that the cell can actually die. And then there are also other kind of inhibitors and monoclonal antibodies that serve as drugs targeting different pathways. So we'll look into some of these hallmarks in more detail. So here is sustaining proliferative signaling. So these are caused by activating mutations in oncogenes. So for example, by gene amplification or gain of function mutations. So here is the proto-oncogene that does not cause cancer by itself, but then there could be mutations like translocations where it has acquired a new promoter. So now it's producing more and more product that might give rise to cancer. So there could be gene amplification. So there could be tandem duplication of the genes or amplification. So it produces more and more protein that might lead to cancer. And then there could be point mutations within the gene or within the promoter or regulatory elements that can actually promote hyperactive expression of this gene. So one of the examples is the BRAF gene, where mutation in BRAF V600E is a very commonly mutated mutation in colorectal cancers and lung cancer. And then also there are a number of tumor viruses that have oncogenes such as Rao sarcoma virus, HIV and HPV that promote cell division and cause cancers. So here's an example of a MYC oncogene that is being amplified here. So there's a lot of product. So the amplification can also be in two ways. One is at the same locus, or they could be as double minute. So here it's amplified as the small regions of the chromosome, so that are called double minutes. Or it could be on the same chromosome itself, called HSR, or homologously staining region. So there are numerous copies of this gene, and then that causes cancer. So these are all the examples of activated oncogenes. So the gain of function mutations in red gene result in autosomal dominant disorder called MEN2, or multiple endocrine adenomatosis type 2, where these patients have a high incidence of medullary carcinoma of the thyroid. So these are again different pathways as to how an activated mutation can cause these proto-oncogenes to produce excess amount or a novel protein that causes cancer. So these are all the list of proto-oncogenes that are present and that are mutated in several of these cancers. So here's a proto-oncogene activated by chromosomal translocation. So this is a very famous translocation called 922 translocation that is present in 95% of chronic myeloid leukemia patients. And this is called as Philadelphia chromosome. So there is 922 translocation, and it is caused by, and it causes cancer because there's two cancer predisposition genes, BCR and ABL, that are now they're actually close by, and then that causes amplification of the of this gene here. So the next one is evading growth suppressors. So these are loss of function mutations in these genes known as tumor suppressor gene. And as I told you, retinoblastoma and P53 are two proteins that activate senescence and apoptosis upon various stresses to the cell. And P53 is a protein that almost plays a role in almost every pathway in cancer. So it promotes cell cycle arrest and apoptosis, and tries to increase DNA repair so that these DNAs that are exposed to all these stresses can be repaired, and then it can be again normal again. But most of these cancers have mutations in P53 gene, and I put a picture of this elephant here because it is known that elephants very rarely get cancers, or um, they almost never get any cancer. So there was a recent publication a couple of years ago, or maybe a few years ago, that showed that elephants have uh, almost tens of copies of the P53 gene. So that is why even if there are mutations in one or two of these genes, these elephants are able to actually, not go into cell cycle arrest or apoptosis, and then they don't get cancers. So the way these loss of function mutations happen is by a two-hit model. This was first proposed by Knudsen. So here you have the two-hit model. So this is sporadic cancers. Let's see what happens in sporadic cancers. So let's say these are homologous chromosomes, and then there's a first mutation, there's a first hit, and then after many years there could be a second hit. So that is why these cancers, they form a late onset. On the other hand, in familial cancers, the person is already born with a mutation, so they've already inherited this mutation from their parents, and then over the course of time they develop another mutation. So these familial cancers or hereditary cancers are early onset cancers. So because the time taken for both the hits, both the mutations, are much lower than compared to sporadic cancers. So sporadic cancers are late onset, and then familial cancers are early onset. So one of the most famous examples, common examples, are in the disease called as retinoblastoma. So if you look at sporadic retinoblastoma, there is a normal gene, RB, and then you require two somatic mutations for tumors to occur. And then these tumors are mostly single tumors, unilateral tumors. So retinoblastoma are cancers in the eye. So usually what happens is these tumors only show up in one eye, because it is it's more than winning a lottery that like you get two mutations, two kind of same mutations in both the eyes. So it is very rare that you get bilateral tumors in sporadic cancers. But on the other hand, there are more familial cases of RB that occur in a Mendelian fashion because of germline mutations. So now these individuals are already born with the germline mutations, and all it requires is one somatic mutation. So the time required here is much longer, but here the time required is much lower. So these tumors are characterized by multiple tumors. There's also bilateral onset of tumors. So these tumors occur in both the eyes, and then these tumors always occur in early onset. Just like we've seen amplification of the proto-oncogenes, here you're seeing the silencing or loss of function of tumor suppressor genes. And it can occur either by epigenetic silencing, mutations, there could be somatic recombination, loss and duplications, or there could be a complete chromosomal loss. So there are different mechanisms as to how these loss of function mutations can occur in tumor suppressor genes. So these are proto-oncogenes and tumor suppressor genes that are listed here. So there's RAS, NMYC, ERBB1, ERBB2, TGF-alpha, SIS, and ABL. So these are all proto-oncogenes. So these require activating mutations. And on the other hand, there are anti-oncogenes or tumor suppressor genes like BRCA1, BRCA2 that are involved in breast and ovarian cancer. Neuroblastoma is caused by mutation in NF1. There's APC or beta catenin that plays a role in gastric, colonic, and pancreatic cancer. And then P53 gene that occurs in majority of cancers. There's also a hereditary form of cancer with mutation in P53 called as Li-Fraumeni syndrome. And then there is a retinoblastoma, and then there is a Wills tumor. So these are all tumor suppressor genes that are mutated. So the next hallmark of cancer is the DNA instability and mutation. So the DNA damage can occur both endogenously and exogenously, and improper DNA repair or mutations can lead to genomic instability. So there are numerous DNA repair mechanisms that play a role based on the kind of damage there is. So if there is just exposure to UV, then there is direct reversal. So there's photoreactivation or methyl group removal. That is pretty quick. And then if there is single strand damage, then it usually goes towards base excision repair or nucleotide excision repair. Or if it occurs in repetitive part of the genome, then there could be like a mismatch repair. There's also double strand breaks. So double strand breaks are caused by non-homologous end joining. There could also be microhomology mediated end joining, or even homologous recombination. So these are three different pathways that repair breaks in that repair breaks in the entire strand of DNA. So if you have a double strand break, you usually get double strand break repair by these three pathways. And base excision repair mostly attends to single strand breaks, even in ER. Mismatch repair, it occurs only in mostly if there are mismatches, that too because of repetitive DNAs. And then there is direct reversal. So another hallmark is enabling replicative immortality. I hope I have here. So this is mainly at ends of chromosomes, which are cap-like structures called as telomeres. So telomeres are made up of repetitive sequences. In humans, it is TTAGGG that occur at ends of chromosomes. And they are additionally bound by telomeric proteins that form a protective cap-like structure. The lengths of chromosomes at the ends are maintained by the enzyme called as telomerase. That is the only enzyme in humans that has reverse transcriptase activity, so that it uses RNA as a template to make DNA. So, so telomeres are almost like the ends of shoelaces, which are called egglets. So egglets are nothing but a plastic cap-like structures that protect the shoelaces from getting degraded. So just like this, our chromosomes also have cap-like structures called telomeres. So if the telomeres are dysfunctional, so then they use the protective cap, and then there could be telomere fusion. So there could be end-to-end fusions, so between chromosomes. And during the next round of cycle, when they divide, there'll be these chromosomes are pulled towards each pole during cell division. And during this, there could be breakage and then additional fusion. And then, these cycles, they keep going on with every cell division. So this is called as a breakage, fusion, bridge cycle. So you can actually see it in many cancer cells. So this is a type of karyotyping called spectral karyotyping, where every chromosome is painted with its own color. So it's either called spectral karyotyping or chromosome painting. So you can see that in a wild type cell, every chromosome has its own color. But if you look at a cancer genome, so these colors are all mixed up. So you actually can't arrange these into different chromosomes. And this has occurred because maybe there is some telomere dysfunction. So there is breakage, fusion, bridge cycles going on, and that is how you can see these. See, for example, here there's a purple chromosome with a red dot. There's a blue chromosome with a yellow region. And then look at this here. There is a blue or purple chromosome with numerous yellow dots. Then here is a blue with red arm totally. So you can never really say what happens in cancer cells. So based on the kind of cancers, there are also different kinds of drugs that you can use. So most commonly they use tyrosine kinase inhibitors. There are so many tyrosine kinase inhibitors in the market now. So based on the kind of tumor, they use different tyrosine kinase inhibitors. Oops, sorry. And then the last hallmark of cancer is metastasis. And then there are numerous pathways as to how even a single cell that that has these genetic imbalances can cause mutations, and these mutations are responsible for metastasis, and then they can go on to infect other regions of the body. So there are numerous pathways that are listed here, and then so this is metastasis here. So there is some cell here that has gone on to have a different mutational profile compared to the rest of the tumor, and that can become metastatic tumor at a different location. So I'm going to end my talk here for the second part. So now that we have discussed like basics of DNA and then the basics of cancer, so in my next part, I will show you what we do in our lab. Thank you.